Welcome to How Ion Mobility Coupled with High-Resolution Mass Spec Enables Analysis of Complex Samples. I'm Tamlin Oliver, Managing Editor of Genetic Engineering and Biotechnology News, and I'll be the moderator for this webinar. If you want to know more about IMMS, Ion Mobility Mass Spectrometry, in case you didn't know, you're in the right place. Our three speakers today will describe recent advances in IMMS measurement strategies in the analysis of complex biological samples in systems biology, proteomics, and protein characterization. They'll also talk about bioinformatic and biostatistic solutions to quickly mine the data collected. As you've probably surmised, they have a lot of material to cover, so I won't waste any time in introducing them and getting this webinar started. Our first speaker, George Stafford, Director of LC-MS R&D at Agilent Technologies, will kick off the webinar talking about ion mobility and what it brings to mass spec. George will be followed by Aaron Baker, Senior Research Scientist at Pacific Northwest National Laboratory, who will talk about increasing molecular coverage in complex biological and environmental samples. John McLean, the Stevenson Associate Professor of Chemistry at Vanderbilt University, will wrap up the presentations with a systems biology case study. The presentations will be followed by a live Q&A, which brings me to my last topic before I let George get started, and that is we want your questions. Submit a question or two or at any time during the webinar, and we'll answer as many as we can upon completion of the three presentations. All you have to do is type your question into the Ask a Question box and hit Submit. Okay, George, I'm done talking. Let's get this webinar underway.
Thank you, Tamlin. This is George Stafford, and I'm delighted to be here this morning. And you can see the title of my talk on the first slide is Development of a New Uniform Field, Ion Mobility Quadrupole Time-of-Flight Mass Spectrometer. And what I want to try to do today is sort of take you through our objectives and how we developed this instrument and how excited we are about the types of information and data that we're obtaining. On the next slide, I'm going to start off here with just some quick acknowledgments because it helps set the stage for the talk. You can see here we have a strong team of scientists, design engineers, and software engineers. And this group represents our Santa Clara site effort. But we also have a strong effort, I'm proud to say, with some collaborators at various universities and laboratories in the U.S. First of all, I want to just quickly mention Pacific Northwest National Laboratory, and as you already know, Aaron Baker is going to be speaking next this morning from that laboratory. But I've really enjoyed our close working relationship with Aaron and Dick Smith at the laboratory, and as you know, they have pioneered the development of ion funnel technology, in particular when it's been coupled to mobility to show the very nice improvements in sensitivity and performance that we'll be talking about today. Also, we've been working with Al Yergi at NIH, and he's helping us to understand the added utility of collisional cross-section measurements to his research. Dave Russell at Texas A&M, and he's excited about the ability to use mobility separation to further his developments at his laboratory. And then I'm also delighted, as already mentioned, John McLean will be talking from Vanderbilt, and he'll be showing some of his most recent results using ion mobility separation. And then finally, we've been working closely with Kathy Costello at Boston University School of Medicine to see how mobility separation coupled with mass spectrometry helps her in her research into complex biological systems. Okay, on the next slide. This is a picture of our Santa Clara team, and I'm simply showing this because I'm sure you're going to recognize some of these people, some of the faces here as friends and colleagues that you already know. Okay. The next slide is an illustration of at least what I think mobility brings to mass spectrometry. And the first, I think the most important, it is a new dimension of separation. It's gas phase separation, ion separation in the gas phase, as opposed to what traditionally is coupled to mass spectrometry, and that's chromatography. As we all know, over the years, as chromatographic separation has gotten better, that's improved our ability to look into complex samples and detect and determine the structure of more and more components in that mixture. And I think what you'll see today that mobility separation even pushes the power of mass spectrometry even further into complex mixtures. Also, we think it's very important to always maintain the best detection limits and sensitivity that you can in any analytical technique. And I'm going to show today how mobility helps this. First of all, it helps to eliminate interferences and background in the mixture. And also, through our development using the electrodynamic ion funnels, how we have very efficiently transferred ions from the ion source through the entire system into the mass spectrometer and detected to maintain the overall system sensitivity. And then finally, I think mobility brings an additional means for confirmation. The direct and accurate collision cross-section measurements is very useful in helping to identify compounds and to correlate ion structures. Okay, on the next slide here, there are 4 points that are listed that I see as our overall or our key sort of objectives and design goals for this new instrumentation. First of all, we wanted to maintain the sensitivity of our mass spectrometers, and we've done this, as I'll show on the next slide, by the use of the electrodynamic ion funnels pioneered in interface with mobility at P&L Laboratories. Also, we wanted to maintain let's say, optimize the performance of the mobility system independent of the mass spectrometer. That is, we've been able to obtain theoretical resolutions of ion mobility and maintain the resolution and sensitivity of the mass spectrometer without affecting either system. We can control the ion fragmentation. We can turn it on or turn it off depending on the experiment that you want to, experimental results you want to obtain. And when we turn off the fragmentation, we can maintain a very low heating of the ions so we don't unintentionally fragment or change conformational structures. And then finally, to maintain the profile of the ion mobility data, we've needed to increase the bandwidth of the data acquisition channel in our instruments, and we've increased this by about a factor of 10 to be able to show the very nice resolution we can obtain with the mobility separation system. Okay, on the next slide, I'm going to go into a little more detail about the mobility part of the system. You can see the figure here now just focuses on the mobility section before the mass spectrometer. I'll go from left to right and sort of walk through the system and talk about the major points. On the far left would be the ion source. I don't actually show the probe, but in this case it could be electrospray or APCI or nanospray. The first thing you see on the left is our heated capillary, and we can maintain our ion source at ground potential just like we do in all of Agilent mass spectrometers. The ions then are sampled into the capillary, and they are drawn by the atmospheric pressure up a potential gradient of about 2,000 volts, so they can enter into the high-pressure trapping funnel. They are shown as the first indicated as the first funnel, or in this case, it's called front funnel. At this point, the gas from the capillary can diffuse and be pumped away while the ions are contained and focused by the ion funnel. They then go to the next vacuum region into the trapping funnel, which is shown there labeled trapping funnel. And at this point, after we collect ions for about 1 millisecond, we pulse these ions out, and then they go into the drift region, and they drift through the gas, and as you can see here, the drift region is about 80 centimeters long, approximately 20 volts per centimeter, and we recommend using nitrogen at about 4 torr. This setup here enables us to achieve a theoretical resolution of about 65 for singly charged ions. Based on this design, again, we've optimized the mobility independent of the mass spectrometer, so both systems maintain their highest performance. And by using the low fields and controlling the AC and DC fields on the funnels, we can minimize ion heating. So if we want to study ion structures and conformations, we can easily minimize any sort of changes to the ion structure. All right, and then finally, by using a uniform low field, we can directly calculate collision cross-sections without using any type of internal ions as calibrates. And then a final major point here, by using a standard collision cell, a CID cell, we can turn we can fragment the precursor ions to produce what we call an all-ions experiment. And what I mean by that is we're able to collect all the precursor ions, fragment those ions in the collision cell, and then collect all the fragment ions. And the fragment ions are separated by ion mobility and then are analyzed by high-resolution QTOF mass spectrometer. Okay, on the next slide, I want to show a photograph of the actual ion mobility system. And the key points I want to point out here is basically the different regions in the vacuum chamber. First of all, starting on the left there again, the first thing shown is the ion funnel, the ion source trapping funnel. The capillary, in this case, is not shown, but it would go into that white Teflon insulator, and so the ions from the source sample through the capillary and then are collected and focused by the first front funnel. Then the next region is where the trapping funnel is maintained. The pressure difference there is about a factor of 10. We go from probably 10 torr in the front region to about 1 torr in the next region. And then finally, the drift region is shown in the third area, and the drift cell itself is actually pictured there in the far right. And then finally in the back is the collection funnel. Again, by very efficiently sampling the ions through the capillary and then using the front funnel, the trapping funnel, and the rear funnel, we're able to maintain very high sensitivity by transmitting most of the ions through the system and finally into the mass spectrometer. Another point here is that you can see that we've taken a lot of care to engineer and design a system that is very simple and is very easy to apply voltages both mechanically and electrically to the system to control the ions as we want as you're performing an experiment. Okay, on the next slide. This slide here just talks a little bit more about separation. As I've mentioned earlier, the mobility separation is a key part. This added dimension of separation it plays a key role in the types of data that we're going to be able to talk about today. And the first point here is based on timescales, you can see how mobility fits very nicely between chromatography and mass spectrometry. Typically, a chromatographic elution is over minutes, where the mobility separation occurs in less than 100 milliseconds. Typically, it's about 65 milliseconds to completely separate the ions in the gas phase in this 80-centimeter long drift region. And then finally, with the TOF mass spectrometer, we can collect a complete spectrum in 160 microseconds. So the mobility fits very nicely in between the chromatography and the mass spectrometry operation without any penalty at all to the duty cycle and the overall speed of the experiment. As an example here, you can see if you were to go into the chromatogram and look at the, let's say, the relatively small peak there circled in blue, that component in the chromatographic elution might actually make up, there may be several different compounds present. And in this case, in this illustration here, we're showing that there would be 3 different ones in that chromatographic peak. We can now, using mobilities, pick one of those. So let's say we select the middle one, and then we can use mass spectrometry to get a mass spectrum of that middle peak separated with in the mobility space. Okay, let's take a look here at a bit of data. Here's an example of 2 sugars that are structural isomers. They are not only isobaric, but they will have the exact same mass, so you cannot separate these based on mass spectrometry alone. And in this particular case, they were infused. There's no fragmentation occurring here, but you can see that they are separated in time by about 1 millisecond in in mobility space. And if you notice, the one that comes out first, the melanocytose, is more of a compact structure. Looking at it, you can imagine how the area or the cross-section of this molecule would be smaller than raffinose, which is shown on the top, and that is in fact the case. It comes out about 1 millisecond later. Notice here, I'll take a moment and talk about what you're actually seeing here in terms of our browser. You can see in this case, we're showing mobility space in the vertical dimension and mass spectrometry in the horizontal. Then there's actually a third dimension here, and that's intensity, and that's shown in the black area in this heat map based on colors to distinguish between the intensity of the ions. And another thing you'll notice here in terms of the mass spectrometry, the mass spectrum shows the identical ions for both species, so the only separation we have in this case is mobility. The M plus sodium is shown at 527, and then the 2 carbon-13 isotopes would be identical in mass spectrometry. If you only had mass spectrometry, those ions would be identical, or that they are identical. Okay, here's an example now of another mixture of 3 ions. In this case, we're using fragmentation. We have 20 volts collision energy in our CID cell, and this is a mixture of these small 3 drugs, ondansetron, colchicine, and reserpine. And you can see here in mobility space, they're very nicely separated over a range of about 12 to 40 milliseconds. But the spectrum in the bottom, the mass spectrum, is a collective drift spectrum, and it includes all the ions. So it would be very difficult to use that spectrum alone to determine, what was present or what was not present. But we can go in very simply, and on the next slide I'll show, we select just the colchicine space. And you can see that indicated here by the yellow area. And now by this picking just this space, you can see here on the bottom the mass spectrum is showing only those ions, in this case the M plus 1 for colchicine. So we can now very nicely identify and pick that spectrum out. And you can go back and do the same thing for the other 2 species as well, the other 2 molecules as well. Now, on this next slide, I just want to show this as a quick example of collisional cross-section measurements and the effect here on the mobility space. You can see, for example, the ondansetron, obviously from looking at the structure of the molecules, would have this smaller cross-section. And in fact, it does. It has a drift time of about 20 milliseconds. Colchicine in the middle, 23 milliseconds, and then finally reserpine coming out at about 29 milliseconds. Now, we made a comparison to some literature values and our experimental or observed values, and you can see that we're getting a very close correlation. They're all within 1% or less. And an example here of how these were calculated is shown on the bottom. We're using the Mason-Shop equation, and the parameters that you need to determine the cross-sections are temperature, pressure, drift voltage or field, and drift tube length. And these are easily measured and corrected in our system, and then applied to this equation to be able to directly calculate those cross-sections shown. Okay, here's another example of 2 small molecules. In this case, it's methylmalonic acid and succinic acid. Again, these are isomers, so they have the exact same mass. And we can separate these in mobility space here. Again, about a millisecond, very nice separation. The mass spectrum here for both of these 2 species is identical, and you can see there in the bottom, we get one single peak at the molecular weight for these 2 species. Now, the example here was shown because these 2 molecules are of interest in the clinical market, and this is something that in some cases to separate these chromatographically has been difficult. So the added dimension of mobility, I think, could be applied here in the clinical market. Okay, now I want to move to a BSA Digest, and in this case, we're now using chromatography. with mobility and also mass spectrometry. So we have all these dimensions added together here. Again, in our browser, I'm showing chromatography across the top from left to right in elution time. Mobility is in the vertical dimension, and the mass spectral data is in the horizontal dimension. And then we have a heat map in the middle showing the intensity of the ions. Now, if you go out to an area in the chromatogram to about 14.36 minutes, you can see that in mobility space here, we have about 7 components that are co-eluting. And you could probably identify maybe the major component from the mass spectrum. In this case, the mass spectrum shown there is a precursor mass spectrum. There is no fragmentation. But in the next slide, I'm going to show here an all ions fragmentation, meaning we're turning on collision energy to about 30 volts. We're fragmenting all those precursor ions and producing all the fragment ions. That total fragment ion spectrum is shown on the top. Now, if we want to identify one of those smaller components, which I think would be fun to try, we picked for example here, we picked the one coming out at about 20 milliseconds. That's the first one. And you can see in our browser, we select that area. And now we can find or just look at the ions in the mass spectrum from this region at 20.26 milliseconds. And that's shown on the bottom. And you can see now we get quite a different spectrum than the total. And we can do a database search of those ions to identify what that peptide is. And that's shown on the left there as a +3 ion with a mass of 435.9 m/Z. So you can see in this example here, it would have been probably impossible to identify this small component in the presence of everything else that was co-eluting at that retention time. Okay, here's another example of, in this case, an HSA digest, again out in the chromatogram at about 14 minutes. But in this case, what we see is we see 2 major peaks, but now the mass spectra for those 2 major peaks are identical, indicating to us that these are probably 2 different conformers that are present at the 14 milliseconds in the chromatogram. And in fact, when we do a search of those ions, again with a fragmentation in this case of 15 volts. So this is an all-ion experiment, 15-volt fragmentation. We do a search of those, and we come up with this sequence here, again a +3 charge with a precursor mass of 547. But we're now proposing that, in fact, those are 2 conformers of that peptide shown there separated very nicely in mobility space. So without mobility, you would have not known that there were a possibility of 2 different conformers present in the chromatogram for this peptide. All right, here's some data on ubiquitin, and this is not showing fragmentation. This is simply showing the ions over a distribution of charges from +5 to +14. If we go in and the browser again and look at the +12 charge state, we can see that it simplifies the mobility space down to the next slide. You can see here that we're showing 2 different conformers for this 12 charge state. The mass spectrum isotopic distribution is shown on the bottom. Now, if we go to the next slide, we can repeat this for all those different charge states, and we can identify the cross-section for each of those charge states. And you can see when you go from a very low charge state, you have a region where the structures are relatively compact. When you go to high charge states, you get into regions where they're elongated or the cross-sections are larger. The data shown here are all generated on our instrument. You can see for charge state 12, you're seeing 2 different conformers. And the reference here is basically for the similar data taken, and comparing it to these approximate regions of compact structure and elongated structures. And then finally, here's a table from a separate reference by Matt Bush showing the ions from charge state 5 to charge state 14, and both our experimental measurements compared to Matt's measurements. And you can see, again, there's very good agreement. We're showing two conformers, in some case, where Matt did not identify two separate structures. And then finally, in summary, I think I've shown that the added dimension of separation brings a lot of power to mass spectrometry. It gives you the ability to look at not only M over Z, but you can look at size, charge, and ion structure of your analytes. The ability to resolve and characterize complex samples using the added dimension of ion mobility to LC and MS, I think is going to also extend the power of this analytical technique over what's currently available. And finally, the ability to measure accurate collision cross-sections, I think, gives it the added dimension of confirmation for your unknown samples. Okay, I think thank you, and I'll stop here.
All right, Aaron, before I let you get started, I just want to remind everyone about the live Q&A session at the end of the presentations. George, Aaron, and John will all be available to answer your questions at the conclusions of all the presentations. So please submit a question now. Aaron, we are ready for you.
Okay. So I'm really pleased to get a chance to talk to everybody today about increasing molecular coverage in complex biological and environmental samples using ion mobility mass spectrometry. So to start off my talk, I wanted to talk about why we're interested in the IMS-MS platforms at PNNL. And so one reason is that IMS adds complementary information to mass spectrometry measurements, which helps lower false discovery rates, it separates isomers, and it allows faster LC separations. We've also seen that IMS helps increase the dynamic range of detection because it slowly allows the ions to hit the detector at different times instead of continuous instruments where all the ions hit the detector at really similar times. And then the TOF doesn't have an AGC, so it has a greater dynamic range than trapping instruments. Our third interest is that we're interested in detecting structural changes in peptides and proteins that could help characterize specific disease states or maybe be determined as structural biomarkers. So the instrumentation we have at PNNL is very similar to the instrumentation George was talking about. One of our main differences is that in our source, we have two inlets where the bottom inlet's used for reference so that we can do on-the-fly calibration for any of our direct infusion or LC measurements. Then the ions are all focused into the high-pressure ion funnel and then into the trapping funnel to be pulsed into the drift cell for IMS separation. Then we use the rear ion funnel to refocus the ions into any mass spectrometer we want to put our IMS front-end separation onto. And there's a picture on the bottom of this slide. One main difference in the ion mobility measurements we do at PNNL is that we do multiplexing IMS measurements. And in traditional IMS measurements, you pulse one ion packet into the drift cell. It goes all the way to the detector, and then you send your next ion packet into the drift cell. However, we found that if we can increase the sensitivity of our measurements by pulsing multiple packets into the drift cell at the same time, as long as you know what time you started your pulse into the drift cell. By doing this, we can also demultiplex the ions into the correct drift time they should be. So if you pulse an ion in at time 0 and then at time 2 milliseconds, we know that the time 2 millisecond ion should have 2 milliseconds subtracted. And we follow all packets and then demultiplex them. We found that this multiplexing approach is very important for increasing signal in LC measurements. And the reason is if you have something really low concentration, you only have a short window of time to get it within the LC. And so if you can cover it 8 times instead of just 1 time, you get 8 times as much sensitivity for that single ion. So this is showing our LC IMS measurements. And this shows the TIC at the bottom for the LC, and then just the drift time packets on the side. So the demultiplexed is on the left, and then the multiplex spectra is on the right. We wanted to analyze how low we could detect ions in these multiplexing measurements. And so we spiked 8 different peptide ions into human serum so they'd be in a complex matrix. And we were able with IMS with a 60-minute LC to detect the peptide at the lowest, or one of the peptides at the lowest concentration of 100 picograms per mil. We weren't able to detect one of the other peptides at that concentration, so we considered that our limited detection with the multiplexing. Then we got rid of the IMS separation, and we just did a 60-minute LC with the TOF MS, and we were only able to detect peptides at 10 nanograms per mil, and we couldn't get any lower for limited detection. So then we went to the Orbitrap, and we know that it's a trapping instrument, so we wanted to give a little longer LC separation so that we weren't saturating the trap quite so quickly. But even with the longer LC separation of 100 minutes, we were only able to detect one peptide at the 10 nanograms per mil level, and then the limited detection was mostly at the 100 nanograms per mil level. So this really showed the power of the IMS and the multiplexing technique. We wanted to understand exactly why we could see concentrations lower with the IMS separation compared to the TOF separation. So we looked at just directly infusing 100 picomolar solution of bradykinin into a QTOF MS and then into the IMS QTOF MS. And if we look for the ion in the IMS QTOF, in just the QTOF measurement on the left, the noise level is actually just as high as the bradykinin level. So it's very difficult. You can see where the ion is, but it's very difficult to say with a 3-to-1 signal-to-noise ratio that you're seeing the bradykinin ion. However, in the IMS, what we saw is that the IMS was able to concentrate the peak into just a small drift time area, and it was able to move the noise to either a different place in the separation, or it was able to eliminate some of the noise because you're trapping the ions, and there's a small amount of heat in that trap, and so it can get rid of chemical noise and get rid of solvent clusters. I wanted to go through a few benefits that we see from IMS that we're not able to see with just MS alone. And one of them with our proteomic studies is that we can separate charge states. And this is very handy for our deisotoping because if we know the drift time that the ion's coming at, we know what its charge states should be. So if it deisotopes to the wrong charge state, we can find out right away that this is a false hit. And these are just showing a BSA tryptic digest on the left and then human plasma tryptic digest. And then compared to George's talk, I put drift time on the x-axis with M over Z on the y-axis, so a little bit of a swap in axes. We also are able to distinguish different classes of compounds with IMS, and this has been very important for complex mixtures. So in this case, we were looking at the difference between peptides and lipids, and we could see that the lipid charge states for the plus ones were different than the peptide plus ones. The lipids are labeled as an L plus, and then the peptides are the P's. And then a lot of lipids are mostly plus one, but you do get some plus two and a little higher charge state lipids. So they really get away from your peptides, which are mostly plus two, plus three, and plus four. Another benefit we've seen with IMS, and George touched on this quite a bit, is that you can distinguish sequence isomers. And so if we had a 9 amino acid peptide and you scrambled its amino acids, you can actually see the difference in the isomers. And in this case, we got one with a smaller structure, one where it's like an S-shaped structure that elongated a little bit, and then another one where it's just starting to form an alpha helix. So we saw a nice separation in these isomers. We're also able to characterize aggregation levels with ion mobility. And on the left side, if we zoom into the red square for heme peptides, we were interested in how many aggregates we were getting in our heme peptides. And if you look on the right side, you can see that we were getting actually monomers, dimers, trimers, and tetramers. And while you can see that kind of in the mass spec, it's very difficult to determine which aggregates are the most abundant and the different amounts of each of the aggregates. So with ion mobility, it was easy to see that the dimers are the most abundant, followed by monomers and tetramers being of similar levels. Another benefit we've seen with ion mobility is that we can study proteins and then bind them to drugs and see if the drugs are stabilizing their complex or causing no effect. So with this case, we used transthyretin tetramer on the left side alone, and we looked and we saw that it has both compact and extended conformations. However, when we bound it to a drug, we saw that the drug actually changed the charge state to a lower charge state, which is a good sign for solution-like conditions, and it got rid of all the extended conformations. You could know that in the mass spectrum, the charge states are changing, but you wouldn't know that the extended forms are gone. So finally, I wanted to talk about a few biological diversity studies and how we're using ion mobility with our proteomics lab. So the important thing right now is that you need to analyze thousands of samples to understand the diversity in a population. And IMS is able to do faster analyses just because you have an additional separation before the mass spectrometry detector. We've analyzed one study where we looked at 3,500 different biological replicates, but currently we're trying to do the bioinformatics to understand the changes we're seeing. So I'm not going to talk about that, but I'm going to talk about another study we just finalized. This study is quite a bit smaller than that study. It only had 120 biological replicates, but we found some very interesting results. And this was related to chronic liver disease with hepatitis C. And it's really important to study hepatitis C because it's estimated in 130 million people worldwide with bloodborne it's a bloodborne pathogen with no vaccine available. In the study, we looked at 60 different patients initially, and they all had liver transplants. Then their liver either didn't have any fibrosis after a certain time period, was a slow progressor and getting a little bit of fibrosis, or the fibrosis happened very quickly, so they were a fast progressor. What University of Washington did is they took and did patient pairs. So if you had a 20-year-old female that had no fibrosis, then they also found a 20-year-old female that had one of the faster progressors. So it was either a fast or a slow progressor, and they paired those together so we could analyze how the pairs had different effects on each other. In the discovery phase, we made it so that you had to find 2 unique peptides per protein in order for a protein to be significant. And from this study, we found 136 different proteins that distinguished between the non-progressors and either the slow progressors or the fast progressors. And you can see from the heat maps below that you can definitely see a change in the different conditions, but due to the diversity of human population, it's not a perfect red to green. So that's why the more patients you can analyze, the better your data will be. The clinicians also wanted us to study non-transplant patients because there's a certain component from just the transplant that could affect the proteins that we were observing. So we studied 60 non-transplant patients with either low levels of fibrosis or no levels of fibrosis to very high levels of fibrosis. And again, we required 2 peptides per protein in order to have a significant peptide. And from this group, we saw 63 patients that overlapped with the conditions for the transplant and the non-transplant, and 76 that were just in the non-transplant or in that group, but with the 63 of those overlapping, so only 15 being not overlapping with the transplant conditions. So this seemed pretty exciting because we saw quite a few peptides that agreed. When we looked at 19 of the proteins from those groups, we were able to find 4 different groups. They were liver metabolism, immune response, oxidative stress, and liver architecture. And we just classified these 19 proteins in this. Some of the other proteins from the 63 we couldn't find functionality for, so it was difficult to classify those. And others could be classified into multiple different groups. So these ones fit the best in these categories. In the liver metabolism, we saw it decrease with increasing fibrosis because the liver's basically dying, and so your proteins there are decreasing. The oxidative stress proteins were increasing, and then we also saw that the extracellular matrix proteins were increasing because you're having the fibroids grow all over your liver, so it made sense that the proteins were going to increase. And then finally, we saw that with the liver architecture, depending on slow or fast, we would see different changes, which made sense because the slow was having less change in the number of fibroids present. in order to verify these proteins that we found, we wanted to look at the 4 different groups, and we looked at 5 specific proteins, and we did Western blots on them, and then compared those to the abundances we observed in the ion mobility studies. And our analyses of these compared very well to each other, so that made us feel more confident in the proteins that we were detecting as statistically significant. So just to end my talk, I have a few slides on future project directions that we've been working on and areas that we think ion mobility is going to be very important. One area is analysis of lipidomic and metabolomic samples. And what we did for this study is we extracted lipids from plasma and then did direct infusion in LC with both the IMS and then just the QTOF MS. And we were able to see quite a few more lipids with the IMS, with the LC attached. And we were also able to see almost the same amount of lipids with direct infusion into the IMS as with LC into the QTOF MS. The reason we were able to see so many more lipids is that we were able to see different isomers at different drift times. And at the very bottom, you can see a single M over Z, and we see 3 different peaks. And what these 3 different peaks correlate to is just a different double bond position for the lipid. And then if you do fragmentation like George was talking about, you can see the fragments below each peak, and you are able to figure out which position the double bond is in. We thought these results were really exciting, so we started looking at smaller molecules, and we were able to see isomer separation for arabinose and xylose, and we saw almost baseline separation for these 2 isomers with a sodium adduct, and the only difference is the position of the alcohol groups. Then we went to more difficult ones for HILIC columns because we wanted to show that we could add information to LC separations. So in this case, we were able to separate ribose and ribulose, and then fructose 6-phosphate and glucose 6-phosphate. So these seem very promising for future metabolomic studies. We finally did an analysis of urine, and we spiked the urine with large amounts of sugar. And so on the left, you can see without spiking the large amounts of sugar, and then on the right with spiking the large amounts of sugar. And you can see the huge changes in the mass spectra on the left and on the right, but we're still able to see almost as many components just because of the IMS separation. And you can see that in the nested drift time spectra. So in conclusion, with IMS, we were able to increase our throughput of sample analyses in our biodiversity studies. Then we're able to analyze difficult samples and obtain more information. And finally, we're able to start running really, really big biological diversity studies. But so far, the bioinformatics have been a little bit difficult to interpret. So hopefully we'll have more results for that in the future. And right now, our future direction, or our current direction, is doing fast lipidomic and metabolomic analyses. And I'd like to acknowledge my group at PNNL in Dick Smith's lab and all of my funding. Thank you.
And thanks, Erin, and thanks for going through your presentation so quickly to put us back on schedule and give us plenty of time for John's presentation and the live Q&A at the end. And again, I just want to remind you about that live Q&A at the end. Please submit your questions at any time for either George, Erin, or John, and we'll answer as many as we can. John, the floor is yours.
Thank you, Tamlin. I really appreciate the introduction. Really appreciate the invitation to speak with you today. George and Erin have both really demonstrated some of the points that I would like to make today, but what I'd really like to convey is our belief in that using very fast separations with ion mobility mass spectrometry, it really provides a tool that's well suited for very complex biological mixtures like those encountered in broad-scale systems and synthetic biology. So when we're talking about systems biology, this is sort of the idea that's put forward by proponents like Lee Hood, suggesting that if we can measure all of the molecular components, both in dimensions of space, but also in dimensions of time, one might be able to sit down like an electrical engineer and be able to work out the circuit diagram of life. And so perhaps at some point in the future, one would go to their physician and she might order a systems biology-like experiment. And then it might be blood, urine, we don't know yet. But then you would come back the next day and she would tell you that, well, we found some circuitry that we need to rewire because you would have developed a particular disease. So what we do for spatial dimensions is a technique that we call imaging ion mobility mass spectrometry. But what I'd like to do is to give a couple of case studies in how we measure the temporal dynamics of molecular evolution and making time measurements. So as a motivational quote, I take this from a 1600s text, and it basically says, the Cartographers Guild struck a map of the empire whose size was that of the empire. And that is to say that it has a scale of 1 to 1. So when we think of a systems biology experiment, do we really need to measure every single molecule both in those spatial and temporal dimensions? And the answer is probably not. But what we need to do is measure as much as we can and go from an untargeted analysis to a targeted analysis very quickly. And so what I'll show you at the very end are our efforts to be able to do just that. One thing that I would note is that our proclivity for generating data is really unsurpassed. We can basically generate about a terabyte of data in an hour. And so what we do with that information in terms of bioinformatics, biostatistics, is of tantamount importance. So George very nicely discussed the timescales of the experiment and how you can integrate ion mobility between, say, a chromatographic separation of the mass spectrometer. And what I would do is underscore the following, which is demonstrated in the lower left. And that is using electrostatic drift cells. One measures this drift time, or what I've called here arrival time distribution. And using the kinetic theory of gases, we can translate that to the surface area of the molecules that you're separating. So what you can do is using these uniform or electrostatic drift cells is to very exquisitely measure the cross-section or squared angstroms of the molecule and interpret those structures in a structural biology way using molecular dynamics techniques. And so the first person to demonstrate this was really Ed Mack in the 1920s. So Ed Mack was a professor at Ohio State University, where what he would do is embed molecular models in beeswax and project light across them. So as he would move these molecular models, what he would do is inscribe a circle on a screen, and the circle corresponded to the rotationally average collision cross-section, or the value of omega, which I'm showing on the right. So even if you have a molecule that exists in two structural forms, like this peptide that I've shown, a mass spectrometer alone would never distinguish them because they're the exact same composition. But one here is a random coil, and the other has more extended structure to it, so it has a larger cross-section. Okay, so this slide shows sort of the evolution of ion mobility in the context of mass spectrometry. So in 1895, Wilhelm Röntgen first discovered X-rays, for which he was awarded the first Nobel Prize in Physics. In 1896, Ernest Rutherford did the first ion mobility experiment, which predates mass spectrometry by about 20 years. So where mass spectrometry really looked at the analysis of increasingly complex samples, ion mobility for a number of years was really relegated to looking at fundamental reaction rates in astrophysics. Probably in the mid-1990s, we started to see the first applications of mobility for the analysis of complex mixtures like those in biology. So now this slide shows sort of a generic arrangement of an ion mobility mass spectrometer. A lot of these experiments have been facilitated by the advances at PNNL and the development of ion funnels. So what we have is the drift tube, which is shown in purple, where you inject ions into this gas-filled chamber, and the separation selectivity is based on the number of collisions with a background gas, which is shown by the little pink dots. So in this region, we want to promote as many collisions as possible, whereas in the mass spectrometer, we want that to be collision-less. So what we have to do is, in between the two, in this interface region, is to drop the gas pressure by about 8 to 9 orders of magnitude very quickly. And so really what you have is an inconvenient marriage, because you want to promote many collisions and mobility and collision-less in the mass spectrometer. But what it allows you to do is to generate a plot like that shown in the bottom left, where you have drift time on the y-axis and M over Z on the x-axis. So this slide shows a typical arrangement of contemporary instrumentation. So in the upper left is electrodynamic high mobility, which is a commercial offering that appeared around 2006. On the bottom left is what I would call electrostatic or uniform field instruments, largely built in individual laboratories, much like our own. And then in the upper right is the implementation by the PNNL group, which was later released by Agilent, which George covered very eloquently. So I'll be showing you data from all three of these types of platforms in this presentation. So what I'm showing here is sort of recasting the timescales that George Stafford showed in the first talk. And what I would like to do is to highlight the bottom portion. And what it does is it shows sort of the order of magnitude differences in the timescales involved. So in chromatography, we're oftentimes dealing with minutes or hours. Then if you're using things like quadrupole analyzers, you're often dealing with milliseconds as well as ion mobility. But what we oftentimes see is the combination of time of flight, which allows microsecond separations. Now, you'll notice that on the far right of the bottom chart, if we're just doing chromatography, you have one chromatogram. But as you increase the nesting of the different separation dimensions, you quickly increase the number of data points that you have. So, for example, it's not uncommon to have something on the order of, say, 10 million spectra. So what's ideal is combining chromatographic separations with these very rapid gas phase separations. We often do that to increase separation selectivity. So what I'm showing here is a UPLC ion mobility mass spectrum, where if we were only to do the ion mobility in the mass analysis, we'd end up with about 5 to 10,000 different analytes for this E. coli cell digest. When we increase the dimensionality with the chromatography, we increase the number of distinguishable features to about 50,000. That's very impressive when we're trying to do our systems biology approaches. Now, this is the reason why we think the ion mobility mass spec is ideally suited for systems biology. And that is, we found empirically that different types of biomolecules adopt structures that are very characteristic of the type of molecule it is. So, for example, at a given mass molecule, if one is a peptide and one is a carbohydrate or a lipid, they separate structurally into these different regions of space. So we can integrate the omics. That is to say, we don't separate proteins and only do proteomics. We do lipidomics, proteomics, and glycomics all simultaneously. So that's illustrated here, where on the left is a model glycoprotein, where we've used PNGase F to knock off the carbohydrate, and then trypsin to proteolytically digest protein. And on the right, what we're showing is lipidomics and proteomics, and this is a human glioblastoma, or brain cancer sample, where you can see a clear distinction between the lipids and the peptides. So by integrating this omics, it allows us to do very complex samples without a lot of sample preparation. So this is also showing a case of so-called isobars, or same mass molecules, where we wouldn't be able to distinguish these carbohydrates using mass spectrometer alone. So what we have here are two human milk proteins where the position of this fucose residue is the only difference between the two molecules. Now, using high-resolution IM mobility, in this case, this is on the PNNL or Agilent platform, we distinguish these two different structural isomers very cleanly. Now, this is a case for lipids. So what we're doing here is using tetraalkyl ammonium salts as calibration standards. We're separating these different phosphatidyl ethanolamines into different charge state families. And now zooming in on one of these regions, you can see the quality of the data here is really quite high. So not only do we have high resolution in the mobility dimension, but then also in the mass spectrometer as well. Now, if you zoom in on one of these correlations, for example, just for lipids, what we find are that these different lipids fall into what we call fine structure. And that is, we can discern even additional details about what type of lipid it is. So, for example, these different color codings represent different classes of families of lipids. Now, turning to systems biology again, this is how we're making temporal measurements. So what we're doing is working in collaboration with a physics group, John Wixlow at Vanderbilt, and an aerospace engineering group at Cornell with Hod Lipson. And we're automating an experiment where we combine microfluidic devices with the fast molecular readout of the ion mobility mass spectra. So we put one of these microfluidic cell trapping devices underneath an inverted fluorescence microscope. You perfuse cells into the traps to load them. And then you use hydrostatic pressure to hold the cells in the traps. Now, if you start putting in chemicals or toxins or drugs into the cell culture media, you can watch all the molecules that the cells release, basically in real time. So what I'll show you now are two very quick examples. One of the first studies we did is to establish a drug test for long-range drug use, and that is when hair, saliva, and urine show you short-term drug use, and that is like a few days ago. The question we were seeking to ask is, if someone used cocaine years ago, would we be able to tell altered biochemistry from the leukocytes or immune cells that they have seen cocaine previously? So what we did is established a rat model of cocaine abuse. And in this model, we had rats put into this device where when they had the stimulus, they could press a lever and take cocaine for about a week. Then they were reestablished on chow for a week, and then they were allowed to take cocaine again if they wanted. And so this is a slide that shows being able to see the metabolites of cocaine at physiological levels in rat serum. So all of these different acronyms correspond to different cocaine metabolites, so we're very sensitive to the physiological levels. Now, when we actually look at the leukocyte responses, we're seeing millions of signals. So what we did is we started developing a strategy using self-organizing maps, which look at the temporal behavior of the abundance of the different molecular features we see to allow us to group into similar regions of space what those different molecules were. And so we term this metabolic dynamics inspector. So looking at the leukocyte molecular expression, each one of these self-organizing maps on the right encode probably about 50,000 molecules. So you can see that when you have naive or non-addicted and addicted, those molecular signatures clump into different regions of space, which allow you to do prioritization to figure out which ones you want to identify. So what we're also working on are a lot of synthetic biology projects. I'm illustrating one here where what we're trying to do is create microfluidic humans on chip. So you would have a brain or a lung, and so forth, where you could do drug testing, but not in an animal model, but rather in primary human cell cultures. So this has received a considerable amount of exposure recently in the press. And what this really allows us to do is look at things like acetaminophen, toxicology, allows us to do drug discovery or disease diagnosis. And it's really trying to get us toward systems and chemical biology approaches. And I hope that illustrates a couple of cases where ion mobility mass spec really helps save the day. I'd like to thank my colleagues who did the work and a number of collaborators whose work was featured here. Thank you.
And thank you, John. Before we start the Q&A session, I want to give the final call on the questions for our panelists. Please submit your questions now. I also want to let you know that a post-webinar survey will deploy shortly in the presentation manager. You might need to disable your pop-up blockers in order to view it. Please do so and take a few minutes and respond. Your comments are extremely valuable to all of us. All right, we are ready to start our Q&A discussion. We have a lot of good questions already, and we'll answer as many as we can.
Okay, everybody, this is John Sterling, Editor-in-Chief of GEN, and I'll be handling the Q&A after Tammy did such a wonderful job on the moderating the presentations. And we do have some very interesting questions, and the first one is going to be for George. So, George, are the fragment ions, once CID is performed, sent back through the drift tube prior to CID?
Thank you, John. No. Once the ions are fragmented in the CID cell, they're transmitted forwards towards the TOF analyzer, where they undergo high-resolution mass analysis. And by the way, the CID it may be selected on or off. You don't always want to do CID, so you may select to have fragmentation or not in that cell.
Thank you, George. Aaron, a question for you, and is how was the quantification done?
So the quantification was done in that experiment at the peptide level, and so we looked at statistically significant peptides to see if they were changing, and then the ones that were changing, we rolled them up to the protein level and then did a protein-to-protein comparison.
Okay, thank you, Jessica. And John, a question for you. Have you seen any fragmentation occurring during IM?
Yeah, that's a great question. So on the earlier home-built instruments, sometimes we would see it, depending on the ion source used, to a greater extent than some of the softer sources we see today. But what that manifests itself as is a streak of signal corresponding to the elution of the precursor ion. Now, it can also, when you want to promote fragmentation or you have a system that does fragment, it allows one to do some really interesting experiments, largely described by Martin Gerald, where you can look at the kinetics of dissociation, and you can look at the kinetics of structural rearrangements, where you see multiple peaks, which are bridged by the fragmentation because of the statistical probability of the molecule fragmenting along the drift tube.
Okay, thank you, John. And George, how is the sensitivity of the ion mobility QTOF compared to the regular QTOF with no ion mobility drift cell?
Okay, thank you. That's another very good question. This has come up many times, and as you can see from the design that all three of us have talked about, we have used the electrodynamic ion funnels to help us focus and transmit ions through the entire ion mobility system. And because of this very high transfer of ions has enabled us to maintain the sensitivity of the instrument, whether it's operated in a QTOF or standard ion mobility QTOF or standard QTOF system. Most of the samples that I'm familiar with and have run in our laboratory here in terms of customer samples or some of our standards, we are seeing extremely very, very similar sensitivities for both modes of operation. In fact, they're so similar, it's very difficult sometimes to tell the two modes apart.
Thank you, George. And Aaron, okay, do M plus H plus sodium ions for the same precursors typically separate in the IM separation?
Okay, and I'm just going to rephrase that just for clarity. What we've seen is if you have a protonated or a hydrogen adduct versus a sodiated adduct, it depends on your backbone structure. So actually of your molecular structure. So if you have a lot of oxygens, they tend to form around the sodium more, so you get a big difference between the protonated plus the sodiated version. But if there's not too many oxygens in your backbone, we don't see too much difference between the plus H and plus sodium version of the molecule.
Okay, well, thank you very much. And John, can separations in gas phase ion mobility be tuned like tailoring the conditions of mobile phase and liquid chromatography? It's a good question.
So that's a very interesting question. So in the 70s and 80s, ion mobility used to go by the term plasma chromatography, but we don't, the separation mechanism is not a chromatography in the sense that you can write a partition coefficient. Now, having said that, you can use the promotion of gas phase interactions like the polarizability of the gas. So if you use an inert gas that's highly polarizable, you might see different separations than one that is less polarizable. There have also been some reports of people using gas additives to also change sort of the selectivity that one sees in the ion mobility, which I think is probably better described as a gas phase electrophoresis.
That was very clearly answered. Thank you very much. And George, is the instrument designed preferably for DIA or DDA experiments?
Okay, thank you. Well, in the design of this instrument, we certainly, with any modern hybrid instrumentation, you always want to leave it flexible so you can do both data independent and data dependent experiments, and that's the case here. Through our instrument control and QTOF operation, currently the modes of operation allow both, and we've followed through here and expect that people will find with ion mobility that there'll be applications that you'd like to be able to have both types of experiments available, and that is the case here.
Well, very clearly done. Thank you. Aaron, is there a method published, application note, about isobaric separation?
And there's actually quite a few nice method papers on the IMS separation. One of my favorites is by Thomas Wittenbaugh, and it's in Topics for Current Chemistry in 2003, and it's volume number 225 and page number 207 through 232, and that's a great one. There's also quite a few others, but that one's a really nice review article on the IMS isobaric separation.
Perfect. Thanks for that information. And John, are ion mobility separations similar to gas chromatography separations?
Yeah, so that's a little bit of a follow-on to the previous question that I answered. I think the answer is no, because in gas chromatography, what you're doing is really partitioning analytes between the stationary support phase and the gas phase, in large part sort of in a selectivity according to boiling point. What we've found is, and we haven't done a lot of studies in this direction, there are some other groups that have, but combining gas chromatography on the front end of the ion mobility mass spectrometer actually affords much higher selectivity and deeper coverage of different metabolites by the separation of chemical noise. So I think GC ion mobility mass spec has a lot of upside potential, in particular in metabolomics.
Good. Those questions were relayed, and I think you answered them perfectly. Thank you. Hey, Aaron, I have a question here. Bear with me. This is a long one. Don't answer. I am trying to purify bacterial toxins from milk and then quantify the toxin using labeled internal standards, online SPE, UPLC, TQ, and MRM. The purification does not seem to work properly, and interference from there's interference from milk components. Excuse me. Would IM help without more purification?
So that's a great question, and we've been doing some research in that area for applications, and not just the bacterial toxins, but we've looked at things where the interference components are really, really high. And so the answer to that question is sometimes yes and sometimes no. It depends if the interference hurt your ionization. So if you use electrospray ionization, if they suck all the ionization possibilities out, then it won't help. But I have seen it help a lot with like soil samples or water samples with high-level interference, where it's a +1 kind of chemical contaminant, and you can move them to completely different spaces in the drift separation. And so like heme compounds or humic acid in soils is a big contaminant, so you can't see as many things in soils, and we move it to a completely different space, and we can pull out probably 10 times as many features as just like a trapping mass spec instrument.
Okay, thank you. And we got one more question for John. What sort of dynamic range does the instrumentation have?
Sure. So the dynamic range, or the concentration dynamic range in the ion mobility mass spec itself, is really about the same as one would get on a mass spectrometer without the ion, simply because we're limited by the detector technology. And so there have been some advances in that regard. But because you have these separations on the basis of different species and the separation of chemical noise, there have been reports, for example, I think David Plummer has shown some of the most, some very compelling data where when you combine LC or GC on the front end of the IMMS, you can increase your dynamic range from about 4 or 5 orders of magnitude to about 7 or 8.
Okay, great, John. Well, listen, unfortunately, we've run out of time, but please note that this webinar will be archived for 6 months on our website, www.genengnews.com. If you missed parts of it, you can watch it again, or you can recommend it to your colleagues and friends, which we highly recommend. I want to say thanks again to the panel for their outstanding presentations, and I want to say thank you to the audience for your attention and your very thoughtful questions about a number of topics brought up during the webinar. And thank you to Agilent Technologies, whose support made this event possible. Thank you, everybody, and have a great day wherever you are.